All right. Just give me a minute. So, all right, let me share my screen first. So first we will be looking at the databases from where we will get the raw data in the form of FASTQ file. So first database is the NCBI SRA. So this is the page of NCBI SRA, and there we can go to, from this drop down menu, we can go to SRA. And here we can write whatever the type of data we want. So in this particular NCBI SRA, we can fetch any application data, like not only RNA-seq, we can take the data exome, whole genome, ChIP-seq, methylation, attack-seq for any application, NGS application. So in this workshop, we are going to analyze RNA-seq data. So we can write RNA-seq. And here because RNA sequencing will be done for many samples, like for animals, plants, anything. So we can put the organism name or a type of data which we want. Say suppose we need ovarian cancer or anything. Say suppose we say cancer. So you can see how many samples it will come. You can see there are total 52,609 samples for RNA-seq with respect to cancer. So if you would like to refine these, search again, because these are many samples. So from here on the right side, there are the list of the organisms. So Homo sapien, Mus musculus, Canis, and so on. So either you can select from here, you can see that for the Homo sapiens, these are the samples. So if you want to select this out of 52,000, so you can select the Homo sapien. You can click here. So now you can see only 37,435 samples are there. So now if you want anything like control, public, so the out of 37,000, these many are the control. It means that you cannot access these many samples. And this is the 35,000 are the publicly available, means open source, and you can access this particular data set. Now, if you want to select whether you want to have the paired end data or single end data, here it is. So single end data, 3,000 samples are sequenced by single end, and paired end is the 37,770 samples are the paired end library. These are the platforms. So if you want these platforms, you can see. And I'm sorry, I have a bit of cough, so in between I might go for 5 minutes. So this is the Illumina platform, ABI Solid. So these are the different platforms. So you can see the maximum samples are being sequenced for Illumina platform. And these are the strategies for which strategies these samples have been sequenced. Yes. So paired end, if the fragment is sequenced in both the direction, single end is only in one direction. So these are the file types. These are the BAM files, and you can see there are FASTQ files available. So you can filter your data based on these filters. So, cancer is again a broad term. Many samples will come. Here you can again specify your search. Say suppose you want ovarian cancer, you can write ovarian. So then data with respect to human ovarian samples will come. So you can see these are the samples. 244 samples related to ovarian cancers are available. So from here, if you want all the 244 samples to be analyzed, you can send these results to run selector. So here you can see these are the RNA Seq samples, and these are the total 244 runs. So here again, there are so many filters. You can filter based on this. Say suppose you want to filter this data on the basis of disease stage or ethnicity, histological type, you can check this. So say suppose I want stage type. So you can see out of 244, 31 are primary, 15 are metastasis, and 1 is not labeled, and 197 are empty. It means that they are not defined whether what stage of disease they are. So if you want to compare between primary and metastasis, you can take these samples. So if you will just click on the primary and metastasis, so now you can see only these samples are here. Total 46 samples are here. If you will move on the right side, so you can see many labeled for this sample. So you can see oncology, ovary. So these are the cell line, library name. So many other information you will get from here. So this is the sample name. This is the material provider who has deposited this data. So these are the cell line, ovary, cell culture. They are labeled. And if you will see, these are the labeled for the type of the cancer. So this is the cancer, carcinoma, adenocarcinoma, mucosal, serous. So these are the different type of ovarian carcinoma. So when you want to make the group of these particular cancer type, so this will help you to make the groups. So carcinoma you can put in one group, clear cell carcinoma you can put in one group, adenocarcinoma you can put in one group, and serous carcinoma we can put in one group. Undifferentiated we can put in another group. So like that we can make the group of these samples. So ethnicity is also given. So whatever the information is given, all you will get from here. So this list you can download from here. Give me a minute. So if you want to download this data, you can download the entire data as well. So we can select all the file or whatever the files we need, that we can select from here. And from here also, when we will select these files, you can see this tab is popped up. So directly these samples from here, we can take it in Galaxy with the metadata. If you will click on this metadata, select it. So whatever it will come, so if you will select, if you will click here in metadata, then the entire data will come. But if you will click here selected, then whatever the samples have been selected, that metadata only will be downloaded. So there are various ways to download this. You can use SRA Workbench also to download this data. So now here we can see there are different tabs. So run ID is there, bioproject is there, biosample. So these are the unique IDs related to this data. Say suppose now I will copy this particular data, project ID, and there is another database within NCBI itself. So now this time select the geo data set. So geo data set is also for the gene expression data. So in this geo data set, you will get both the data set from microarray and RNA Seq. So, and here also you can search with RNA, you can write keywords, RNA Seq, humans or cancer or whatever it is. Now I have copied one project ID. I will put this project ID. So you can see this is the cancer cell line encyclopedia bio project. So this particular project, if you will click, so this particular project is not present in geo data set. But what it is present, we will see. So this is present in SRA, but not in the geo data set. So how to search in the geo data set? So all data is not present in geo data set, but there are some overlap data set which are present in geo data set and as well as SRA. So if we again go to geo data set, again we will write RNA Seq, and again we will write cancer, ovary, human. So if we will search this, you can see these are the data set present. 651 samples are present over here. So there are some samples which might be common. There are some samples which will be different. So here now you can see for this particular fifth number, it is present in both. So you can send to SRA run selector. So from here also you can see this particular project is also present in SRA run selector. So from here you will get to know which project is also present in SRA. So you can see this is also present in SRA run selector. So suppose, and in this particular data set, you will get some processed data, not the raw data. So say suppose if I will click on the first item. So you will get the all experiment details over here, what experiment has been performed, platforms, which platform they have used. So they have used HiSeq and NovaSeq. And if you will click, so in this particular project, there are 131 samples. These are the labels of these samples. So you can see these are the labels. So this is the genomic and immune landscape of long-term survivors of high-grade serous ovarian cancer. So you can see the project ID is same. So here you, sorry, you won't get the FASTQ files, but you will get the expression count. So this is the, you can see all raw data text.gz. So from the XTP, you can download this data. And this is the total processed file. So this is the correlated log to fold gene. So here you will get the differential expression count matrix itself. So you do not really need to process this data. So if you want to start filtering and looking at the gene expression after differential gene expression analysis, then you can take it from here. So, and in this, these are the unique ID. Like in SRA database, SRA is the, so this is the main ID for this database, SRR number. In geo dataset, it will be labeled as GSM ID. Now there is one more database. This is called ENA. So ENA is the ENA browser, European Nucleotide Archive. So once we click over here, the same project ID, we can put it over here and then see view. So the same project will open over here. So you can read more about this particular project, what they have done, and who has done some primary information you will get. And you can see all these samples raw data files. So you can see these are the raw data files fastq.gz. And in this particular project, there are 4,550 samples. From here, you can change the view, how many samples at a time you want to see. So you can see how many samples are there. So you now here also you can see the project ID, SAM number, SRX number, SRR number, which you can go and match from here. So these three databases are interlinked, and from these three databases, so except Geo database, ENA and NCBI SRA, you can fetch any type of NGS based raw data FASTQ files. Right. So now the second thing is that we need reference databases. So for reference databases also, there are many databases. So let us have a look from where we will get the reference genome. So again, the first important database is the NCBI. So now this time click on all database and click on genome. And here you can put your organism name. So once you put that and then click on search button, then your genome page will open. So it will give you the information, what is the current genome assembly. You can download this reference in the FASTA form called genome, transcript and protein. You can download the annotation file in different file formats, GFF, GenBank and tabular. You can do the blast against these data. So whether genome, transcript and protein directly from here also. And you can browse whatever the genomes are used to construct the assembly to reference genome. So these are the, you can see the annotation report, you can see the organelle annotation report and some information about the taxonomy and other related information about the organism. If you will come here it is given the summary of your sequencing data. So this is the genome assembly generated from these many human subjects. These are the sequencing reads. You can see the genome assembly read clicking on it. So total length is this, protein count. So in the human genome, these many proteins are present. And this is the GC content. So if you will come, related publications are also here. And these are the chromosome wise FASTA file. So if you are, if you need, if you do not require unknown sequences and the mitochondrial sequences, you can click on these files, then you will get chromosome wise file, and then you can merge them and make your own reference genome from here. Or otherwise, if you do not want to do, you can just take it from here. So you can click, once you will click here, these FASTA files will be downloaded. So if you need genome, click on genome. If you want RefSeq reference, you can click on transcript. If you need the protein, you can click on protein. So this is how you can search the genome in NCBI database. So now another database is the Ensemble. So this is the Ensemble genome. Click on Ensemble genome browser. So then this page will open. So from here, you can see what all species reference genome is present. And then you can access human, mouse and zebrafish from here also. So say suppose you want human, you can go here. So here you can see different file formats. So if you want gene, cDNA, non-coding RNA, you can download from here. This is the annotation file in GTF and GFF format. If you want the FASTA file reference, then you can download from here. Download DNA sequences. So this is the genome reference. So from here also you can do that. The another database is the UCSC. So don't go to the university page of the UCSC. Click on the UCSC genome browser. So once we are here, we can go to download. So here also there are different utilities. You can use MySQL, you can use the cloud access, you can install REST API, you can download through FTP. So there are many things. So if you will click on Genome Browser Store, so from here also you can download. But this will be through the Linux or another databases you have to install and then use it. So basically, go back and then go to genome data. So source codes are also given over here. So these are the organisms which you want to download the reference genome. So go to the table browser. So from here you can set the parameters and then download the reference genome. Say suppose here you want the genome. Here you can define your regions, or you can go to the all the chromosomes. So from here you can select mammals, which particular order they belong to. From here you can select the organism, which assembly you want. So always try to take the latest assembly. So the latest assembly is GRCS38 HG38. So what you want? So mapping, sequencing, you want custom track, you want genes and gene prediction. So whatever you want, you can look at it. So then track, which track you want. So these are the tracks you want. So you can take that. Then table. So you can define your tables as well. Otherwise, select the default. Genome. So look up. Then you can create the file intersections. And from here also, once you select your files and the options, from here also you can check this option if you directly want to send it to Galaxy. So once you will select that and click on get the output, then it will directly go to Galaxy also. So this is how you can download. If you do not want to download the Galaxy, just uncheck this and then get the output. Then it will download. So another important database is so this is UCSC Ensemble and NCBI. The another database is the iGenome. So iGenome Illumina. So iGenome is the portal from Illumina. So this particular website is maintained by Illumina vendor. So here you can see there are different organisms, and all the reference genome from different databases are also present over here. So what is the advantage to take it from here? So in this particular folder, so this is not just the genome. If you will download this particular folder, in this pre-indexes will also be there. So you do not need to index your genome before the alignment. So that is the advantage over here, and it is more frequently updated because this is maintained by Illumina vendor, and on their website portal, when you have the access of the data analysis, they will also use the same reference genome for the analysis. So this is done. So this is the reference genome, and there were some questions. Yes, we were searching for the RNA sequencing. What does it say? RNA sequencing 47. So how many projects are present? Different type of projects are present. That will tell you in the sequencing strategy. So this was the question, right? In this strategy. Yes, so this strategy, how many different type of experiments are there? So different projects will be there. So in that, how many different projects are there? So all right, let me just now for what we are going to do in this particular hands-on sessions for 3 days. So what we are going to do, we will go to ENA. So we have already one project. So I'll keep that session ID here and see. So this is the data from the Illumina HiSeq 1500 RNA Seq of ovarian cancer ascites cells. So here you can see this sample, and then if you will click on the project, how many total number of sample in this project you will get. So in this particular project, total 24 samples are there. So you can see all 24 sample. Now here you can observe that for the one sample, so this represents one sample. So you can see only one file. So this is the single end sequencing for this particular project. So if you need more information about this project, you can copy this project ID from here, come back to NCBI. So you can check where all this information is present. So first again, go to SRA, put this project ID. Then go to this particular project. And we are here. So you can go to an SRA experiments. So these files are not present in the SRA selector. So if you will go to geo dataset, so you can check in geo dataset as well. Put the same project ID and search for it. So we are here. So again, it is coming over here. So these samples, this data is not present in the individual files are not present. So if you want, you can select from here also. So biosample and the SRA. So all related experiment, you can see again, go back and see what is present. So, but here you will not get the raw data. I will show you some other way also. So even you can go to run selector directly. So here say suppose you will keep SRA run selector. So this is the SRA run selector. Click. Then we are on SRA run selector. So suppose we will keep the same project over here and search. So you can see this file will open over here. These are the individual files and you can see the information from here. So directly sometimes they will not give the tab over there, but if you will go to SRN and selector, it will come over here. So you can see these are your sample labels. So these are the tumor associated macrophage, tumor cells, tumor associated T cells. And if you will come down, these are the tumor associated macrophage. So in this, what the researchers have done, so they have compared the gene expression profiling between the ovarian tumor cells and tumor T cells and versus tumor associated macrophages. So this is how they have done this experiment. And if you want, you can again check this ERR number. So this ERR number is same over here. Right? So here, if you want 20, 21, 22, you can see what labels they are. So this is you can see 20, 21, 22. So these three samples, which group they belong to? So they belong to tumor associated macrophages. So in this particular, from this particular project, we will take the three samples because we need the replicates. We will take these three samples for the tumor associated macrophage, that is one group, and we will take the three samples tumor cells. So we will also try to compare the differential gene expression between tumor cells versus tumor associated macrophage cells. So we will take three samples, so that these 3 samples will be one replicate and these 3 samples will be another replicate in another group. So we will make our group and we will do the analysis, differential gene expression analysis, tumor cells versus tumor associated macrophages in ovarian cancer, which is associated with the ascites cells. So our sample number will be 20, 21, 22. So these will be 3 samples, and then tumor associated cells, if we will go up, so this will be, so 31, 32, 33 we will take as a tumor cells. So what is missing here in ENA? The data, the samples are not labeled over here, what these sample files are. So sample file label we will get from NCBI. So you will get, there is no data set which is available in SRA and not available in ENA. All the samples which are present in ENA, the associated metadata is present in SRA. So everything, and these are the IDs you can map from here, from SRA to ENA and vice versa. So now we need these samples. So 20, 21, 22 and 30, 31, 32. These samples we will take. Now how to take these samples directly to Galaxy? Say suppose now we will open our Galaxy. Just give me a minute. All right. So how to take these samples directly? So when you will open your Galaxy account, so this is the home page of Galaxy. So once you enter your login details, you will land up on this page. So on the extreme right side, you can see history. So this is the project history. You can create your project. You can see the files. You can see the data. You can see the output. You can create the input for your data set. So from plus symbol, you can create new projects and save the new projects. So what is the benefit over here? Your projects will remain same in your account. No one will delete until unless you will not delete. So there is a time limit. Before that, only you can delete. No one else can delete. Even the Galaxy team who maintains this database, they also cannot delete your files without intimating you. So if you have many projects using this tab, you can switch to different projects. You want to name this particular project, you can click on pencil symbol and here you can write. Say suppose this is the RNA Seq ovary and then press enter. This project is renamed. How to get the data directly from here to our Galaxy? Say suppose we are here, so you can see this tab, generated FASTQ files FTP. So if say suppose you will click this file, whatever the data you want, now you can see, you can this tab will pop up, download selected file. If you do in this way, then these files will download in your local system. But we do not want to download and upload again, so we will take directly to Galaxy. So to do so, on this particular file name, copy link address. Come back to Galaxy. Here they in the tool under the toolbox on the extreme left, there is a tab called upload data. So once we will click on upload data, this window will open. So if your files are already in the local system, because this is we are taking from the database, but if you are generating your own data, it will be in the local system, then they will not be in this database. So you can go to choose local and select directory and then upload your data. If you want to take it from some other FTP site, then you can do it from here. Choose remote files. So FTP directory is there, your Dropbox is there. So there are multiple things you can take directly. If you want from FTP, NCBI FTP, Ensemble FTP, you can also choose these databases directly. But we want these data from ENA database to Galaxy. Now you go to paste and fetch data. Then this window will pop up. in this box, in this search box, we can paste this link and press enter and come to new row. In this, we can put any number of links, any number of sample data which we want, but one link per line. So again, come back here, copy the link of another file. If this data is the paired and data, you have to take both the files, not only one file. Just that you have to remember. So this is the one group 20, 21, 22. These are the tumor cells associated with macrophages. And another is the 30, 31, 32. We can go back and see again. Yeah, 31, 32, 33. So from here, 31, 32, 33. So when we copied all the links, we can say start. Then this window will become green, and at the back end, you can see all the 6 files which we are going to analyze. And you can refresh this either from here or from here. When these files, these tabs are in pink in color, it means that the process is going on. Once the process is over, then this will become green. So now you will be wondered how this data is being processed. So this is the GUI window of your account, but at the back end, these things is happening on the Linux terminal. So this is just the GUI of your terminal. Here, this is the so many tools are present in this toolbox. So if you want to use this, because there are many tools over here in each tab. So genomic manipulation file, convert formats. So these are the individual tools. So before you go and proceed for the analysis, you should know what tool you want to use. So you can put your tool in the search box, and then this tool will open over here. So say suppose now we want to use this tool, we click on this, and then the options will opened over here. You can select the options, you can select the input file, you can select the output options, and then execute the command. So this is how the Galaxy works. There are some data which is already available in the share data library. So go to share data and then go to data libraries. So if you want some data is already there in the Galaxy, so you can see what data you want. So say suppose you want some data, you can keep it over here also. And this is the all the genomes and annotation files are also present in this Galaxy. So I'm using Galaxy Europe. These genome and annotations are not, this folder is not present in Galaxy India. This is present only in Galaxy Europe. If you will go to histories, so this will show you if you have shared your histories with somebody else. So these are the some predefined histories which are being shared by another people. If you want to see the workflows, so there are some pre-configured workflows are present in Galaxy. So these are the, here also you can put any search what you want to analyze, and then it will give. So these are the all the workflows, and based on these ratings, you can choose the workflow. So you can see who has created and how many, what they have created, and if somebody else is using the same workflow, they will rate it. So based on that also you can filter. So there are different things which you can do it over here. You want to come back to your home button, you can click on home. Then this will show you the current history. So every process, every step takes some time. Don't think that on a single click everything will happen. No. Whether you use here, whether you use something else, but at the same time, say now this process is going on. This is already going on the server of Galaxy. At this time, even if you will shut down your system, you will log out from your account, this process will keep happening. And later on, if you will come back, your data will be downloaded in your account directly. So till this process is going on, you can unmute, ask any question is there so far. And also in the Galaxy, when you use the tools and some anything, there are some pre-configured genome references are also there for some common organisms, well annotated organisms. You can use them. If you do not want to use them, you can come over here and then similarly download sequence FASTA format. Say suppose we need genome in a genome reference. Again, we can right click here, copy the link address, again upload data, paste and fetch. If we will put here, we will say start and then close. So similarly, you will have the reference over here. But I'm not going to download and upload here because for human, the reference already the genome reference is available within Galaxy. So if still I want to use my own, I can take it from here. Genome and annotation file, I can take it from here. So genome and annotation. So you can see Homo sapiens HG38. I can select this and go to export to history as data set. From here, I can select my history, in which history I want to put this annotation file and then click on import. Then if I will see, so already my annotation file is here in the same history. So there are multiple things which you can do that comes with your experience. And when you know how many tools, what the data is present and what we are going to use. So all right. So you can see these are done. So if you want, so now there are many tabs which are popped up over here. So this button is to delete the file. If you want to rename these files or give some label, you can edit these attributes over here. So say suppose we want to label these files for our reference so that we can give, so this we can consider as T1. So this is the replicate. So this is T1 means treatment 1. If you have many groups, you can label them accordingly and then save it. So I'm not going to relabel. I will remember because this is a very small data set. So I'm not going to change this. If you want to visualize what is there inside the file, how the files look like, how the data looks like, you can click on I icon. then your data will open. This file will open over here. So you can see how this sequencing file looks like. So this at the end, if you see 1, it means that this is the forward file. If you see the 2, then it is the reverse file. So for the paired and data, when you will look at, so then this will be labeled as _1, 220 _1. So if it is _1, then it is a forward file. _2 is the paired file. That is the reverse file. But this is only single and data. So we will get only one file. So the first line starts with @ and this is the unique identifier. This is the sequencing line. This is the plus symbol. This is the quality header. And fourth line is the quality of each base. So now you can see these are the characters, right? So when we will do the QC, it will convert these ASCII letters into a quantitative score. So here when you visualize, this is just I am saying, but this is not a good way, but you can see that in some reads, this first base is the N. Just remember this. So when we receive the data, any data, raw data in this file format, then first thing is that we have to do the QC of the data. If we are taking this data from even the database, we do not know how this data looks like. So we have to do the QC on this data. So we will be using fast QC to do the QC. So this is the fast QC. Click on multiple data set. Then your all the files will be here. Click on the empty space and select all the files. And then scroll down and then execute the tool. Then it will run the fast QC. It will generate 2 files per file, per sample data file. It will generate 2 files. One is the graphical report in the form of HTML and another is the tabular, just the raw data. So now we would like to, so this is the individual report. So how many reports if we have many more samples? So these are the 6 samples. So we have so many files generated. Say suppose now we have many samples, 50 samples, 100 samples. We would like to compare between these samples how the data looks like. So then from the fast QC data, it can generate the consolidated report. So for that, we can use multi-QC. So multi-QC aggregate results from bioinformatic analysis into single report. So here there are many options. If you are running those tools, like if you are running BAM tools, if you are running BCF tools, Bismarck, Botai, so it can give you the consolidated report. So we want from fast QC. So this is the option fast QC output. So output of the fast QC report. So we want to use the raw data files to generate the consolidated report. So from here you can select only the raw data files. And then I can execute. So here I can queue the commands, and because for this particular multi-QC, we need the output of the fast QC. So once this fast QC will be over, then multi-QC will start. So before we go into more step of the analysis, first we have to look at the quality of the data. So that is very important. So as a first step, once you get the data, run the fast QC to generate the quality report and look at your data, how the data is, whether data is good, data is bad. There are different parameters on which the data will be evaluated. And all these files will not be give you the output at a time, because for each sample the data is different, the amount of data is different. So based on how much data it is there for each sample, it will process. So you can see for some sample it is being done. So let us have a look. So once this will become green, it means that the sample processing is finished. So if you would like to see the how the data looks like, you can click on eye icon, then this report will open. So these are the parameters on which the raw data will be evaluated. Basic statistics. This will tell you the file name, file type, encoding. So how these ASCII letters converts into quality score, which encoding algorithm it will be used. So there are two algorithms, Sanger Illumina and the Solexa. So it will use the Sanger Illumina encoding. How many total number of sequences are there? If any sequences for poor quality, it will flag. So there are two parameters, individual base and the entire string of the sequence. So this will show you the entire string, not as the individual base. This is the sequencing length. So this is a 51 base pair, single end data. Percent GC, 56%. So if you will click on per base sequence quality, so you can see the per base sequence quality. Here on the x-axis, these are the number of bases or number of position of the bases. So 1 to 51, because this is a 51 base pair sequencing. On the x-axis, this is the quality score from 0 to 40. So this 0 to 40 is divided into 3 zones, pink, orange and green. This pink zone is up to Q20. If the data quality is equal to or less than Q20, then this data is not good. If the data quality score is in between 20 to 28, it is optimal quality. We can enrich and reach to the green zone. So there is a possibility for this, if the data falls in the pink zone, then you will lose more data when you are trying to enrich for the good quality. And most of the time it won't happen also. So if your data quality is 20 or lesser than 20, better to regenerate the data. If the quality score is above 28, it is a good data, and we can see our data is good data. For this particular sample, still we have to look for other samples. So per sequence quality tile, as we discussed yesterday in the theory part, the Illumina platforms use the flow cell. The flow cell has the lanes. The lanes are divided into tiles. So these tiles are represented over here. So this is not related to really our data, but this is more related to our instrument and the machine. So if repeatedly you see in your data any particular tile, any particular position and in the lane is not giving the good data, then you can check the instrument settings or the flow cell cleaning and other stuff, which will be taken care by the sequencing team who handles the machine. Per sequence quality score in this particular thing. So these are on the x-axis, this is the quality score, and on y-axis, these are the number of reads. So initial these many reads, what is the quality? Next these many reads, what is the quality? And here we can see at the end, what is the overall quality of the data, that is the 39. Per base sequence content. So these are the number of bases. This is the position 1 to 51. And for each position, what is the content of ATGC in all these sequences? And then these are the number of AT and GC. This is the percentage. Then the percent GC content. So here you can see the GC content of the data. So ideally what GC content tells us, the GC content tells us whether your data is coming from one genome or not. Because every individual genome will have a specific GC content, and then it will be represented over here. If the data is coming, so this is the theoretical distribution. Blue graph represents the theoretical GC content. So if it is a single peak like this, it means that your data is coming from the single genome. If your data will follow the similar kind of path on the blue line, then it means that your data is fine. But if you can see many peaks or it is going here and there, it means that you have some contamination with your data. So we know that this is a cell line data. So maybe in cell line some contamination is there, or this is not very distinct peak from blue color line. It is not away from this blue color graph. So maybe this might be for some gene which are very overexpressed, or maybe some small contamination you will be able to see. But when we do the analysis and other stuff, this will go. There are steps to take care of these things. So per base N content, how many N's are there in your data? So we have seen visually that in some reads it is N, but it will calculate the overall N's in the data, and based on that percentage it will be represented. Maybe in few reads N's are there, not in many, so that is permissible. That's why here it is showing the N contents are not there. But if N's are there, that also can be trimmed and removed. Sequence length distribution. So we have seen this is the 51 base pair data. So here it is showing 51 base pair. Duplication level. So this is the gene expression levels. Definitely we expect some duplication because of the overexpression of the genes. Overrepresented sequences. So again, when you have the overexpression, some reads will be overrepresented. So this will be represented over here. But at the same time, if they are not belonging to other genome, so it will show you as no hit. It means that this data this overrepresentation is from the same genome which we are dealing with, not from any other contamination. Otherwise, it will show you if it belongs to some other data, it will show you here. Adapter content, this is the most important parameter in this our QC report. You can see at the end, if the adapters are there, it will show you as a sigmoidal graph at the end. So there are, you can see some colors over here on these tabs. So green means all the parameters are good to go. Red color, take care of the parameter. It is not good. Orange color is permissible, but you have to just be careful when you are doing the analysis. Again, there are among all the parameters, some you can ignore even though if they are red color, but if there are some parameters, if they are in red color, you cannot ignore. Say suppose if my adapter content is in red color, I cannot ignore that. I have to take care of this. If my GC content is red, I need to look into it, because if I'm dealing with the data where I expect the data coming from only one single genome, then it cannot give you multiple peaks. I need to get only one peak. So there are some applications like host pathogen interaction applications where you can definitely see multiple peaks or the metagenomic analysis, because there you will have the more than one genome. N's, definitely you cannot ignore the N's in your data, because if many N's are there, your alignment, or if you are processing without a reference, that is the de novo, you are constructing the assembly. Then if you have many N's, then your assembly will be fragmented or your alignment will not be perfect. So looking at these parameters and the reports, you can take care of it. So now multi-QC is run. So these are the individual sample report. So you can look at it, but if I want to see how the other samples are there together, I can click on this report page, multi-QC web page. So this is nothing. This also looks like this, but it will be, so this is for one sample. This report will be for all the samples. So now you can see parameters are same, which we had looked at into the fast QC report. So you can see now here, all the samples are here. The percent duplication, GC content, and the number of million of sequences are here. So this is in number of reads. This is in the percentage. So this is count. This is percentage. And here you can see these are the light blue color are the unique reads. Black color are the duplicated reads. This is for the quality. And more specifically, we want to see whether adapter contents are there or not. We can click over here. And here we can see, yes, there are some samples which have the adapters. So we need to remove these adapters. So to remove the adapters, we have a tool called trim glue. So what the tools we are using for the present hands-on? These are not just only the tools. There are many tools. If you go through many papers, they will all the researchers might use different tools. You can use any tools. To use any other tools, you need to understand the basics of the tools, how they work, and what the parameters you have to set. So for to do so, you can go to Google. If you would like to use some other tools, you can read the manuals and then get into that how to use those tools. So this is the trim glore. So to trim also, there are many tools which you can use, but you need to get experience, and that experience comes when you will use keep using this and keep practicing all these things. So trim glore is there, quality and adapter. So once we click over here, the files will be here. So if the paired end data is there, you have to select the paired end and you have to select both the files together. Remember this. This is very important for the paired end data. If you have single end, then we have to choose single end. We can click on the multiple data set, click on the files, and then if you know the adapter sequences, you can select the user defined adapter sequence. And if you know which library preparation kit is being used, you can select this. So Illumina Universal Adapters list is there. It will be there. So this is the database data. So we do not know what they have used, what is this adapter sequences. So we will click on the automatic detection and then just execute. So this will remove the adapter sequences from our data. Also remember, and this will give us the filtered reads. So now if we are doing the filtration, the downstream analysis, alignment and other things we have to perform on the trimmed reads, not on the original data. And then also remember, once we trim the reads, then we need to again perform the QC to look whether our data is get rid of adapters or not. So that is also important. Don't think that we have run we trimmed the reads, then the adapters are removed. We need to be ensured before we proceed for the alignment that yes, adapters are removed. So then this will remove the adapters from the data. So we can even further QC. Now this trimming is going on. We can queue the next command. Say suppose again we will be using fast QC. So this time we have to select all the trimmed reads, not the other reads. So we can select only the trimmed reads. 1, 2, 3, 4, 5, 6. And then execute. This will be executed once the trimmed reads will be done. So we can queue the multi QC also. Multi QC. In here we have to write the fast QC and then insert fast QC output. Now this time we have to, whatever the after trimmed it, we get the raw data that we have to select. So once the trimmed this parameter will run, then fast QC will start. Once fast QC will be done, then multi QC will start. Because for these for these tools, the input is the input from this will be the input, and for the multi QC, the input from the fast QC output. So even though at this time also, if you will log out and shut down your system, nothing is going to happen because you have already executed the commands on the server. So this will keep running. If you will come back after 10, 15 minutes or 20 minutes, everything will be done. So let this finish. It will take for a while to run this. So if meanwhile, if you have any question, you can ask. You can take a break of 5 minutes and then come back. Yes. These are not the primer sequences, these are the adapters. Because I want to ensure that the adapters are not there after trimming. So it is important to run the fast QC again. So you can see for the samples which trim glow is being done, even the fast QC is started and that is also done. So this is how this will be, this will run. We did not align yet. First thing what you have to do, you have to check the quality of your data. If the data quality is not good, then why you will go for the alignment? It is the unnecessary wastage of time as well as the computational resources. So that's why it is very, very important when you, as soon as you receive the data, you have to check for the quality. Yeah, for doing the QC, we do not need reference. Uh, and this tool you can use in any application data generated from genome, transcriptome, epigenome, anything. Yes, the ASCII on this ASCII data. So, and now you can see that this all trimglore has run, and for all these fast QC has started. It will take a while to finish this, and once fast QC will be done, then it will generate the consolidated report using the multi QC.
Welcome back to my channel. My name is Sarah.
So now we can see as soon as the fast QC is over, multi QC started by its own, and it will take a while to finish this as well. All right, if we will look at the consolidated report, so we can see, we can compare this data from the earlier multi-QC data and see how much data is being removed. So looking at it, you can see how much data was the trimmed and then enriched for the good quality data. So now you can see it reached to from 39 to almost 40. Because our aim is not to enrich for the good quality, because the quality score was good, but just to trim the adapters. So N contents, adapters, duplication labels, overrepresented sequences, adapter content. Now you can see no sample found with any adapter contamination greater than 0.1%. So this is a good thing that we have removed the adapters from the data. Now we can proceed for the alignment. So before we decide that whether we have to do the via alignment or via de novo assembly, we need to check whether our organism for which we are dealing with the data that has the reference genome or not. So this is a human reference data. So RNA seq data from human cells. So we have the reference genome for the humans. So we will be proceeding for the reference-based alignment. Now this is the RNA seq data. So here when we do the RNA sequencing, only the exons will be sequenced. Other intergenic regions or intergenic regions and the introns will not sequence. So only the exonic part will be sequenced. So when we align our exonic data to the DNA reference genome, it will go and align only the exon. So introns will be there, but it will not align. So there will be a gaps between exons. So we need to use the gapped aligner or the splice aware aligner, which knows the where the splice sites are there. So to do that, there are different tools, there are different pipelines. You can use star pipeline, you can use top hat pipeline, you can make a combination of it and then use it. So depending again on your data, on your computational power, and what type of analysis you want to do it. Because from RNA seq also there are multiple type of analysis which can be performed other than the standard pipeline. So standard pipeline will standard analysis will be concentrating on the differential gene expression analysis. Other things what you can do the fusion gene discovery, you can do the novel transcript, you can do the splice analysis, you can do the variant calling on the same data. So there are many things which can be done. But again, we will be using the HiSet tool for the alignment to genomic data. So HiSet is just for the alignment. If you want to do the more different type of analysis, better to use some other pipeline like star and top hat, because there are some options where you can use for the fusion genes and other things. HiSet 2 less options are available. So here our main aim is concentrating on the differential gene expression analysis. So we will be using the HiSet 2. Also, this is the fast aligner as compared to top hat 2 and star. Star is very computational intensive resource, so you need very high computational power. to use star pipeline. So for this workshop, we will be using HiSet2. Our data is not very big data, so and it will be processing fast. And this is a sensitive aligner. So we will use HiSet2. We will be using the built-in genome. So as I showed you, you can take your own genome also from the human, from any database, and then keep it use. If you are doing that, you can choose the from history, but I will be using the built-in genome. So here I can put human, then we will be landing up with GRCS38, HG38. So always use the latest genome for the analysis. So what is our library? Our libraries are the single end library. So here we can execute all the files together also. So this will give the individual BAM file, align file. So for that, keep the multiple data set and select the trimmed file. Don't select the original files, because now we will be dealing with the trimmed files where adapters are not there. Select all the files. We need these. We do not have whether which strand this library is prepared, whether plus strand or minus strand. So we can give the stranded. In the summary option, we can give the print alignment summary. So this will give us the alignment summary for each alignment. What is the total alignment percentage? So this is very, very important to do the QC. There are tools also, but this will just print the summary. But if you want genome-wise coverage, genome-wise alignment summary, there are tools to generate the QC report. But here we will be just first, as a first step, we will print the alignment summary and then execute. Then we will get for each file, each sample, we will get the aligned file and the mapping summary separately. So this will alignment takes time. So that's why I hold it for a long, because today I wanted to finish this alignment. So by tomorrow or maybe in some hours, this alignment will be done. So tomorrow we will be looking at how to visualize these BAM files, and then the next process, how to generate the count files, count matrix, and then the differential gene expression analysis. So that is it for the today. If any question, any doubt is there so far, please let me know. So, all right, there are no questions. I will stop the session over here, and we will meet tomorrow at 6 morning. Thank you so much.
Recording stop.